Hello. So these are the notes we took either Wednesday or Thursday, depending on whether you're A or B day. We're going to start with 4.10 because the last one we did was 4.9. Most of this you already have in your notes, and we've already talked about in relation to other things. But I'm just going to run through it real quick. If you want to jot something down, pause the video. Otherwise, you just need to worry about copying down the highlighted stuff on the next few slides. All right. So the process of meiosis is similar to mitosis. Mitosis just makes a copy of something like a skin cell. Okay. Meiosis is kind of like doing mitosis twice. So you end up, instead of with 2 cells, you end up with 4 cells in meiosis. And each of those cells has half the chromosomes of the original cell. So these in males become sperm, and in females they become eggs. Now, so when they join, they form a zygote. Remember, zygote means joined. Okay, so they join and form a zygote that has 46 chromosomes, like the average human. Okay, now if during meiosis some of the chromosomes don't separate like they're supposed to, then you could end up with a sperm or an egg that has too many chromosomes or too few chromosomes. One example is if a zygote is formed and it has 3 copies of chromosome 21, then that's Down syndrome. And the people with Down syndrome are mentally disabled. They have physical differences. Klinefelter syndrome, they have normally genetically. Now look, y'all, I told you before, I ain't worried about gender. That is a you decision. Okay. Be happy with yourself. Be kind to other people. That's all I can ask. But genetically, because we're talking about genetics, genetically speaking, a male has an X and Y chromosome. A female has 2 X chromosomes. Sometimes, if again, these X and Y chromosomes don't separate properly during meiosis, you might have someone with 2 X's and a Y. So genetically speaking, they would be a male, but they would have elongated limbs. I know it says organ on the slide. I can't change it right now. But I believe it's supposed to say elongated limbs, and they can have learning impairments. Okay. Most embryos, though, that have too few or too many chromosomes don't make it full term of the pregnancy. They don't survive. So, but there are some cases, and there's a few other cases where these things can happen. You don't need to know specific examples. What you might need to know is you might see a question that says a child is born with 3 copies of chromosome 21. Okay, which of the following is the type of mutation that happened? And you would say that it was a chromosomal disorder. Okay, so that's the kind of thing you might have to answer. I'm not going to ask you to define Down syndrome or Klinefelter or anything like that. Okay. So after reading 4.10, going over those notes, you should be able to tell me what chromosomal disorder is caused by an extra chromosome 21. That's Down syndrome. What are the sex chromosomes for a male and a female? So a male, genetically speaking, is XY. A female, genetically speaking, is XX. What happens to most embryos with extra chromosomes? Okay, they don't make it. They end up as a miscarriage. All right, there's a video from the New York Times on Dolly, the clone sheep, but it's like 13 minutes long, so I don't think we'll be watching that. 4.11 is on cloning. Again, if you're taking notes, you would only copy down the highlighted material. So cloning is the process of making an exact copy. Okay. All right. And if you look at the little bubble with my face in it, there's a plant. This is a succulent called stonecrop. And you can actually take one of these leaves. If they break off, I wouldn't pull them off on purpose, but if they break off, you can put them in some moist soil and they can actually root and form a whole nother plant. Now that whole new plant is going to be genetically identical to this plant because the cells are produced by mitosis. There's no sexual reproduction involved. So that's asexual reproduction. The A means not or without. So asexual reproduction means that there's no trading of sex cells. Now, bacteria can do this. Plants can do this. There's even a few animals that can do this. Starfish, if they are cut in half, some starfish can regrow the other half. And then you have two starfishes. There's actually a story about some oyster farmers that wanted to get rid of starfish because the starfish were eating their oysters and cutting into their profit. So they're like, hey, we got to get rid of these starfish. So they took a big metal net and they drug it across the seafloor where the oysters were and chopped up the starfish. And they're like, we got those starfish. Yeah. So what they did is they cut up the starfish, and then some of those starfish fragments were able to regrow the other half. And so now they had at least twice as many starfish as they did before. They made the problem worse. If only they had had a good 8th grade science teacher. Ding, ding, ding. Okay? Look, I know you're like, I'm never going to use this in real life. You never know when you might need something. Those fishermen needed to understand that starfish can reproduce asexually by being divided in half. And if they had understood that, they would have known, chopping them up was not a good idea. Okay? You never know when you might use science. All right. So bacteria can do it. Some single-celled organisms can do it other than bacteria. Plants can do this. Not all plants can do this, but a lot of plants, you can take a cutting and you can put it in some moist soil and keep the soil moist, and it will sprout roots, and you have a whole new plant. Some plants, like strawberries, you have the plant, and they'll send out a little modified stem, and that little modified stem will go across the ground, and then it'll get to a certain point, and it'll start putting down roots. And then a whole new plant grows up beside the original strawberry plant. Okay. And then on the next slide, I have a video about Hydra that I'll show you. 1997, Dolly the sheep was cloned. That was the first animal that was successfully cloned. And it took 300 embryos before they got one embryo that worked. Okay, so cloning is not super efficient. We're not making the clone army anytime soon. Okay? All right, so let's see here. Now this slide is a video. So what I'm going to do is I'm going to play the video. The speaker is going to be on my the projector, and I'm going to point my microphone at the projector. So hopefully you'll be able to hear this. If not, I'll have subtitles on. Well, that should work.
Hydra, a freshwater polyp, belongs to the phylum Coelenterata. Microscopes have revealed an interesting act of budding in Hydra. Budding is an asexual mode of reproduction which is accomplished quite rapidly. Somatic cells divide to form a protrusion or swelling on the surface of the body called a bud. Let us see how it all happens. Nutrients accumulate at some point near the middle or in the lower half of the body. The cells of the epidermis and gastrodermis divide and form an extension called bud. Gradually, the cylindrome extends inside. Tentacles also appear as protrusions in the center of which the mouth develops. Once the bud attains a certain size, a constriction arises at its base, and the little hydra starts feeding. Eventually, the bud pinches off from the parent. It is said that the bud may help in separation by holding on to an object with its tentacles and then pulling itself away from the parent.
All right, so hopefully we can still hear. And so that's a hydra. They're related to like jellyfish and stuff. And they're really, really super tiny. But it's kind of cool. It's an animal that can reproduce asexually. Okay. Let's see here. So after reading 4.11, you should be able to define cloning. Bacteria and plants can clone themselves. This form of reproduction is sexual or asexual. That should be a question mark. So you should be able to tell me that cloning is asexual. What was the first cloned animal? Dolly. When did it happen? 1997. Does cloning always work? No. All right. This is another video. I'll let you look this one up yourself. There's the link, and it's about DNA and the making of RNA and how RNA helps make a protein. But it's not necessary right now. You should have seen it in class. All right, 4.12 human genome. So the Human Genome Project was started in the late 90s, I believe. And the idea was they were going to sequence the whole human genome, all the genes in the entire human species. And they completed this in 2003. 3 billion bases they sequenced. The trick is that those 3 billion bases make up like 1 million different codons that make up like 20,000 genes. All right, so they're still analyzing some of those genes and figuring out what exactly they do. And, it's been estimated that a lot of our DNA doesn't necessarily do anything. It's just there, left over from, evolution and whatnot. All right, some of the benefits that have come out of the Human Genome Project: we have tests for disorders now. We can test genetically. We can test an unborn fetus, and so the parents can plan accordingly. If there is a genetic or chromosomal disorder, we can test people, like adults. Some chromosomal issues or genetic disorders don't show up until later in life. So we can test those people maybe before they, want to start having children, to see if they could might pass on something like Huntington's disease or sickle cell disease. Okay. New technologies for sequencing genomes. That's important when we're studying animals and bacteria and all sorts of things. And knowing your own personal risk for having a genetic disorder. All right. Now, this slide, this is from my personal 23andMe test result. So according to 23andMe, I'm 68.4% British and Irish. Okay, 11% French and German, and 5% Scandinavian. So what that tells me is I'm white. Okay, all the commercials, everybody's like, oh, I found out I'm Native American. Oh, I found out I was related to Abraham Lincoln. Yeah, nope, I'm just a white guy. So, but I'm okay with that. Doesn't make me any better or any worse than anybody else. I am just who I am. All right, now this is some of the genetic tests they did, because I did the Ancestry and the Health. So, for example, Parkinson's disease, those variants are not detected in my genome. So it is unlikely that I will have Parkinson's. Now, I could develop something like Parkinson's if I had brain injury or something like that. But genetically speaking, I'm unlikely to develop Parkinson's disease. Celiac disease, again, those variants are not detected, which I can eat gluten. Okay. By the way, I don't know where you are on that. Celiac disease is a gluten intolerance. That is an actual disease. Okay. Now, some people decide to not eat carbs or to not eat gluten because they just make that decision. Some people, honest to God, can't eat it because they have celiac disease. It's a genetic condition. So if someone tells you they're gluten intolerant or they can't eat gluten, be kind, because they may have celiac disease and they maybe can't help it any more than someone with a peanut allergy or, asthma, right? Type 2 diabetes. I have the typical likelihood. So my interpretation of that result is that I don't have any genes that make me more likely to develop type 2 diabetes, and I don't have any genes that make me less likely either. So basically, what that tells me is I need to exercise more and maybe watch what I eat a little bit, because being overweight is a lifestyle choice that can affect my chances of developing type 2 diabetes. Okay. So that is something that is me. I got to worry about that. But genetically speaking, I am not at a higher or lower risk for developing it. All right. So, and there were actually a lot more genetic tests they did. And thankfully, most of them, variants not detected. So, yay. They must be Levi's because they're good genes. I really hope someone watches that because that was clever. Maybe not funny, but it was clever. All right, you should be able to define genome. After reading that section, you should be able to tell me what the Human Genome Project accomplished, and you should be able to tell me the benefits of a Human Genome Project. All right, we're going to skip that one. 4.13, gene therapy. Gene therapy is the insertion of genes to cure a genetic disorder. So take someone with type 1 diabetes. That's where their pancreas just doesn't make insulin, right? Now, so with type 1, there could be a genetic disorder where they just don't make the insulin. So if we could take the gene for making insulin and we put it in bacteria, okay, then the bacteria will make insulin for us, and we can just collect the insulin, put it in a bottle, and people can use that. However, let's go a step further. Okay, what if we took a virus that the only thing a virus does is inject its DNA into cells, and then that DNA hijacks the cell and makes it make more viruses? But what if we took that virus that's so good at injecting DNA into cells, and we take out the virus part, the DNA that's bad, we throw that away, and then we take the gene for making insulin, and we put that in the virus, and then we use the virus to inject the gene into cells. Then the cells would have the gene for making insulin, and then they would start making insulin. Okay, that's gene therapy. And they do this with different kinds of cells. I think they've even tried it with bone marrow to correct sickle cell anemia. But some studies, some cases, it has worked well. Some others, they've had to repeat the process because it worked for a while, and then they had to repeat it. So it's promising. I don't think they've perfected it yet, but it's a promising idea to help a lot of people with genetic disorders. The only thing is, let's say you have a genetic condition like sickle cell disease, and let's say you undergo gene therapy and it completely cures you, great. You won't pass that on though. If you have children, you will pass on the sickle cell trait if you have it. So that's something to consider. It cures you. It doesn't prevent you from passing it on to your offspring. All right, so after that, you should be able to tell me what gene therapy is for which kind of disease. It's for genetic diseases. It's not going to cure cancer. It's not going to cure the common cold. Okay, it's not going to cure COVID. It's only for genetic diseases. What do they use to get the new gene into cells? They use a virus. Okay, all right. So here we have another video. I'll let you look this one up if you want to. It's about genetically modified foods from the Royal Society, and they're supposed to be a kind of an independent. They don't specifically support genetically modified foods, and they don't specifically denounce it either. So they're in the middle. They're independent. And they say that they report only the fact, okay, of the matter. So it might be worth looking that up. All right, 4.14, agriculture. So biotechnology is a big part of agriculture these days. They took corn plants and inserted a gene from a bacteria that produced an insecticide. So the bacteria naturally made an insecticide, something that would kill insects. They took the gene for that protein that was the pesticide. They put it into corn plants. So now the corn plant leaves would produce this insecticide. So then when bugs come along wanting to chomp on the corn plant, they die. Okay, that saves the farmer time and money of having to spray insecticides. It means the farmer is going to get more crop per acre of land. That's called yield. Okay. We can also modify crops that maybe they need less H2O. So now the farmer is using less water. The farmer has can maybe grow in places they couldn't grow before. Okay. So there's a lot of benefits here. Another example, this isn't agriculture, but another example of how biotechnology is useful. They used to get insulin from cow or pig pancreases, and that's how they got insulin for people with diabetes. Okay. Then they figured out how to insert the human insulin gene into some bacteria. And so then they just got bacteria sitting in a petri dish, gobbling up some food and making insulin. Okay. How crazy is that? These bacteria just sit around all day making insulin. All the technicians have to do is collect it. Maybe they filter it or something, and then they put it in a bottle and send it out to people who need it. Okay. I'm sure that made insulin a lot cheaper and easier to get, even though for some reason it's still expensive if you don't have insurance. People should have insurance, like universally or something. I don't know. Anyway, so that made things a lot simpler. Gene therapy may even go a step farther. Gene therapy is another type of biotechnology. All right, so after that unit, you should be able to tell me what biotechnology is. Bio means living. Technology is using knowledge to solve a problem. So biotechnology is using our knowledge of living things to solve a problem, like how to make insulin. Transgenic crop. Trans means across. Gen means gene in this case. Or origin. So transgenic means that you're an organism that has genes from a different organism. So for example, one of the first genetic experiments they did of transferring genes is they took the gene that makes fireflies glow, or lightning bugs, whatever you want to call them. They took that gene and put it in a tobacco plant. And then the tobacco plant made the same material that made the firefly grow, or glow, and then the tobacco plant glowed, in the dark at least. Okay, so that's cool. What are some benefits of biotech? Medicine. Technically, the process of making yogurt, fermentation, that's biotechnology. Ethanol production is biotechnology for your, fuel for your cars and stuff. Biofuels, that's biotechnology. Having to water your crops less, having to fertilize your crops less, having maybe being able to grow a crop somewhere they normally wouldn't grow due to the lack of H2O. Adding nutrition to plants. There's places where all they have is rice to eat, and so they get enough calories to stay alive, but it's not nutritionally is not nutritionally enough. It's missing nutrition. So they can add nutrition like vitamin A to rice, and they call it golden rice. And the idea is that the rice will then not only provide calories to keep people alive in these places, but it will also add vitamin A, which is important for children's eye development. You can, if you're a child and you're growing, your eyes are developing, you can actually go blind from not having enough vitamin A when you're little. So it's really important. So biotechnology is a big thing, and there's lots of different branches. If you're thinking about careers and you're interested in medicine or farming or just about anything really, even if you're just worried about solving the energy crisis, I mean, biotechnology is everywhere. So take a look at it. All right. And then here's another video you can look up if you so desire. We'll probably watch that on the next day. All right. Have a good day.